So today we're going to start a deep dive and discussion into DNA, which really is going to be the link that's going to hold together the rest of our semester. So remember, in the S phase of the cell cycle, the copying of DNA was a crucial part in order for mitosis to occur because DNA is so important. And it's also was so important that when it got too stressed out, the cell will actually divide to relieve the pressure on the DNA molecule. So now that we've kind of learned about its importance, let's start to actually study the molecule itself. So today we're going to have a discussion over the topic of DNA structure, where the essential question that you should be able to answer by the end of this lecture is, what is the structure of DNA in humans? So first off, to start off our discussion about DNA, we need to know what is DNA. So DNA stands for deoxyribonucleic acid. And so you can see where the DNA comes from. It's going to be found in the nucleus of all eukaryotic cells. Prokaryotes also have DNA, but it's circular and it just floats around in the cytoplasm. Generally, DNA is going to be in the form of chromatin, that thread-like stuff that we saw as we studied our cell cycle and mitosis. It does, though, condense into chromosomes. A complete copy of this DNA is found in all cells of the body, and segments of DNA code for traits that we refer to as genes. So let's dive right into what is the structure of DNA. Well, the structure of DNA was actually unknown for quite a while. And it actually wasn't a published known thing until 1953. So there was a lot of science that went into this, and there was a huge push to understand finally what that molecule of heredity actually was. So the structure of DNA was discovered by James Watson and Francis Crick, as mentioned in 1953. In fact, those two gentlemen received a Nobel Prize for their work with the structure of DNA. And in order to get there, though, there were multiple players in this race to understand that structure of the last macromolecule that we have to really deep dive into. So these gentlemen had help from Maurice Wilkins and Rosalind Franklin, as well as Edwin Chargaff. And so all of these players were involved in the large puzzle to put together all of the little pieces in order to understand the structure of DNA. So clue number one in this puzzle was actually figured out by Edwin Chargaff. And Edwin Chargaff was able to take multiple samples from multiple different organisms. And what he did was he looked for the presence of the four nitrogenous bases that are found in DNA. And so he measured the amount of each of the different bases in all of those different samples and species. And he was able to see a very clear pattern that led him to publish his work on this. So looking at the sample data table below, do you see the pattern that Chargaff saw? Well, hopefully what you noticed was that the amount of T always equaled the amount of A, and the amount of G always equaled the amount of C. And so this became known as Chargaff's rules, and Chargaff's rules then led to the base pairing rule, which says that A will always bind to T and G will always bind to C. So in regards to this, there will always be the same amount of adenine as thymine because they bind together or they match up, and there will always be the same amount of guanine and cytosine for that same reason. So let's go ahead and apply our learning. So if we look at the following table, it's asking you to fill in the amounts of each of the different bases present in sample Q and sample Z. So take a minute, pause the video, and figure out what you think each of these samples equals. Then resume the video, and we'll look at what your answer should have been. So with sample Q, we see that we have 15% adenine. That means that we are also going to have 15% thymine. So how do we go about and figure out how much guanine and cytosine is present? Well, if I take 15% and add it to 15%, that gives me 30%. So I know that all four of these have to equal 100%. So if I take 100% minus my 30%, that leaves me with 70% that has to be represented by guanine and cytosine. Since guanine and cytosine have to have equal amounts, we divide 70 in half, and it becomes 35% of both in sample Q. Sample Z is a little bit more simple, but the same process applies. If I have 25% guanine, that means I have 25% cytosine. 25 and 25 gives me 50. 100 minus 50 gives me 50. So that means my adenine and thymine will also be 25%. The second clue that led Watson and Crick to actually be able to determine the structure of DNA is they had a picture of DNA taken by Rosalind Franklin utilizing X-ray crystallography. This photo became very famous and was dubbed Photo 51. And in this photo, it clearly showed the sugar phosphate backbone of DNA, it being a double helix, and the nucleotides or bases being in the center. So putting all of that together, what is the actual structure of DNA? Well, there are several different aspects to the molecule of DNA, but first and foremost, it's made up of repeating subunits called nucleotides. We studied this back in first quarter when we looked at the macromolecules model that we had. And so a nucleotide itself is made up of repeating parts. And so in order for a nucleotide to function, it has to have three components. The first is it has to have a phosphate. It has to have a deoxyribose sugar, which is this pentagon. And it also has to have a nitrogenous base. And that's going to be your ATCG. So the structure then, when these nucleotides get put into place, actually makes it like a twisted ladder. And so the ladder, because it has two sides with rungs in the center made up of our bases, but then it's a helix shape, so it turns in on itself. So again, the rungs in the center of the molecule are made up of the bases pairing complementary, and the sides of the ladder are made up of alternating sugar and phosphate molecules. If we then look at how all of this is held together. So let's put all of this information together. The sides of the DNA are held together by phosphodiester bonds between the alternating sugar and phosphate molecules of the nucleotides. So that means the nucleotides are flipped and alternate back and forth between the sugar being on the outside or the phosphate being on the outside. The bases then pair complementary in the center and make up the rungs of our ladder. So the complementary base on one side pairs with its complementary base on the other side. So you need to remember your base pairing rules here of ATCG. The bases themselves, though, are held together with hydrogen bonds. Where have we seen hydrogen bonds before? Well, you're right. Water. And so much as water molecules were held together by those weak attractive forces of hydrogen bonds, actual DNA itself is held together by some of those weak attractive forces called hydrogen bonds. So let's complete the strand. If one side of the DNA is ATA, TCA, TGC, GGG, what would the second side be? Pause the video, go ahead and figure out what the second side would be, then resume the video to check your work. Very good. The second side would be TAT, AGT, ACGCCC. Now I need you to take a minute and I need you to draw this entire structure as pictured here into your notebook. Please take time and use colors. While you don't have to match these colors exactly, everything that is colored on this DNA molecule must also be colored on yours. You will notice in this picture that we have things labeled that we haven't yet discussed, and that's okay because we're going to be using this modeling or this image as a piece that we go back to when we talk about replication of the DNA, and those things that we haven't talked about yet are going to become important in the replication conversation. Once you are done copying this, please resume the video. Okay. Next, we need to ask ourselves how all of this DNA can actually fit into each of those tiny little cells. So every cell in your body has just over 6 feet of DNA if we unravel it and measure it from end to end. Your cells, however, are so small that you can't see them without being aided with a microscope, and what's inside of them still remains unclear with the level of technology. So it's very important to understand how DNA is able to be packaged so efficiently and still able to be used. And so when we look at this, the largest structure is going to be a chromosome, which in that condensed form, remember, you can see in a microscope. If I start to take that chromosome, though, and unravel it, you will see that it goes into supercoils. The supercoils turn into coils. The coils into nucleosomes. The nucleosomes are made up of histones and a string of chromatin, and then the chromatin itself is made up of the double helix. If we watch this as a video, let me restart it for us. You can see that same process of taking a chromosome, unwinding it. There's the supercoils, the coils, the nucleosomes, the chromatin, down to the double helix of the DNA. It is important that you understand this structure and how it works in order for DNA to be packaged. And so besides understanding the previous slide's diagram, I do want you to copy this information into your notes, and you can see how every structure is shown as it progressively gets from the smaller to the larger structure. Once you're done copying that data table into your notes, resume this video. So this brings us to the end of our notes on this topic. And so go ahead and answer your essential question in the summary section. When you are done summarizing your notes, go ahead and let's check your learning and make sure that you can answer each of these questions. Thank you.